Hey everyone, a growing body of evidence links gut health with longevity. Atop that list is fiber fermentation to produce short-chain fatty acids, or SCFAs, but there's a lot more to that story. To dig deeper, today's guest is Dr. Dan Weiner, who will illustrate gut bacteria-derived metabolites that not only positively impact health, but also negatively. So welcome, Dan, and let's get into the presentation.
Thanks, Michael, for giving me the chance to talk today. I'm going to talk about gut metabolites, but through the lens of the gut immune system. And the reason I do this is because the gut immune system is one of the main responders to gut metabolites, and the gut immune system has a profound effect on on the body, both from in terms of physiology and how inflammation is controlled through the body. As a main example of how this occurs, I'm going to talk today mostly about the gut immune system in obesity and insulin resistance. This is highly relevant to aging. I say this is a great, model for most chronic diseases of aging. And when thinking of the gut immune system and how it controls chronic disease, this might have been the first example of how it works. And it's been popping up in many other chronic diseases of aging more recently, such as neurodegenerative disease. Just one conflict of interest related to this. I'm a co-founder of a company called Propion. We make prebiotic formulations, and we use also gut metabolites to treat and also for the goal of promoting healthy aging. Well, because we're going to talk about diabetes and obesity as an example, and give some background for the people so they can learn about it. There's two broad types of diabetes. Type 1 diabetes is a traditional autoimmune disease, which is characterized by your immune system, particularly these T cells called CD8 T cells, killing off the beta cells of the pancreas. Type 2 diabetes, which is our focus today, is not considered to be a traditional autoimmune disease. However, it is characterized by this low level of inflammation in the body, which predisposes to something called insulin resistance. Insulin resistance is when the tissues in your body don't respond as well to insulin, and which means insulin can't act well on the tissues, and that leads to rising blood sugar, and I'll get into that. Both genetic and environmental factors contribute to obesity and type 2 diabetes. But in terms of type 2 diabetes, by far the biggest risk factor is obesity. It's important because people with diabetes are at risk for many different types of vascular complications. So these people are at risk for things like heart disease, stroke, problems with their eyes, their kidneys, nerves. It's one of the leading causes of death in the United States and Canada. Last time I checked, I think it was seventh. Oops. Okay, so it's a big problem. About three-quarters of adults in the United States are either overweight or obese. Obesity is there's a number of ways to define it, but I mean the most common is still body mass index, which is basically the amount of weight per height. And so a BMI over 30 is considered obese. The prevalence of obesity is increasing not just in adults, but it's also increasing probably more in children than adults right now. I mean, adults, it's almost plateaued. It's about three-quarters, as you can see, but it's going up in children. And as I mentioned, it's a widespread issue. About 9% of the population in the United States has diabetes, and we spend like an enormous amount of money trying to treat it and its complications. So the question today we're going to talk about initially is like obesity related insulin resistance. So how does obesity lead to insulin resistance in the body? So it does so through there's a number of ways that obesity contributes to insulin resistance, but from an immune point of view, it predisposes to this low level of chronic inflammation in metabolic tissues. And so one of the main tissues we look at is visceral adipose tissue, but it also contributes to inflammation, other things like liver and muscle. And a new upcoming thing that we've uncovered over the last 10 or 15 years that obesity causes this state of altered gut immunity in the intestine. And this is where we'll see how it links to the metabolites. Okay, for people who aren't that familiar with the immune system, there's two broad groups of immune cells. There's this part of the immune system called innate immunity, and then there's also adaptive immunity. So innate immunity, these are the types of immune cells that kind of go around and they kind of gobble up anything that looks dangerous. And they recognize, very broad patterns and things like bacteria. And the main types of cells would be like macrophages, dendritic cells, and neutrophils. These were ones that I might talk about. Neutrophils are like, and they respond very fast. Like if you cut your hand, you get this redness, inflammation, and you might get some pus. That's like the neutrophils coming in and kind of like degranulating and trying to kill off bacteria. Then usually like days later, what happens is the adaptive immune system kicks in. These are T cells and B cells. And basically, these innate cells, such as dendritic cells, will talk to the T cells and cause them to become activated in the presence of a virus or a bacteria. And then these T cells are the orchestrators of the immune system. The adaptive immune system recognizes, very specific patterns in bacteria or viruses or even self proteins, for example. B cells recognize them in these things called antibodies. T cells recognize them through a T cell receptor. And then when these T cells get activated, they make these chemical messengers called cytokines. And I'm going to be talking about a lot of cytokines in this talk. That's how the T cells communicate to other immune cells, but also to other cells in the body. And usually, there's a number of different types of cytokines, but usually if I say something like interleukin, that's a cytokine, that's a chemical messenger. Okay, so because fat is one of the tissues that gets inflamed during obesity and diabetes, we'll just go quickly over the types of fat in the body. There's two broad types of fat. There's brown fat and white fat. Brown fat dissipates energy as heat, whereas white fat will store energy. White fat then is broken down into visceral fat and subcutaneous fat. Visceral fat is the deep fat in the body that connects the internal organs together. So that would be like, connecting, stuff, between the intestines or in the liver and around the kidneys that you can see. It's a subcutaneous fat is the fat that's deposited just under the skin, and that occurs mostly in and around hips and can build up like around hips and other places in the body. Now, the visceral fat is thought to be the type of fat which is most adversely impacted metabolically. And this is the fat that typically gets more inflamed. And so what happens is when somebody is lean and has healthy fat, many of the immune cells that are in there are producing these cytokines. You can see it, IL is for interleukin. And these cytokines are usually anti-inflammatory cytokines. And the most common anti-inflammatory cytokine would be something like interleukin 10. So they also produce, to a lesser extent, some cytokines that are associated with this thing called type 2 immunity. Type 2 immunity would be, an immune response responding, like in the presence of a parasite or like in some allergic conditions. And these cytokines, are thought to be also to maintain homeostasis in lean adipose tissue, but by and large, it's the anti-inflammatory ones like IL-10. And it does this. I don't know if I've got the slides on that, but it does this by maintaining proper signaling of the insulin receptor. Now, when people become obese, what happens is the adipose tissue, and we're still in visceral fat here, so that would be visceral adipose tissue. The fat cells expand as you get obese, and they expand to the point where they can compress on the surrounding blood supply or they become stressed themselves. And then what happens is they start releasing chemical messengers called adipokines, and that's from the stress, and that brings in and recruits immune cells. Now, these immune cells come in and they start to interact with the fat cells, and ultimately, as this continues, the fat cells become stressed and they start dying off. I mean, I use the term necrotic here, but that's probably not the accurate form of cell death. There's a whole bunch of forms of cell death, and it's not really known exactly the way adipocytes are dying right now, but it could be from a number of different types of cell death. But these immune cells come in, they kind of clean up this mess of the adipocytes, dying off and getting bigger and stressed. Now, if you look at adipose tissue in someone who's obese, about half the cells are, immune cells in there, and that includes things like macrophages that become inflamed. That's what these big red things are. And then these macrophages will make cytokines, and then these cytokines will be things like inflammatory cytokines, almost like it's responding to a bacteria. So like interleukin-1, interleukin-6, TNF. These things are important because they act on the fat cells, and what they do is they jam up signaling downstream of the insulin receptor, and that contributes to insulin resistance in fat. So that's how the immune system contributes to insulin resistance. And so when fat becomes insulin resistant, a number of things happen. It doesn't uptake sugar as well. It can't turn free fatty acids into triglycerides as accurate as well. And so what happens, free fatty acids can leak out and get into the liver and other tissues. And so that kind of kickstarts many aspects of metabolic disease. But when I started my lab, this was probably back around 2010 or 2011, we were interested what causes this inflammation and fat. So why does the fat become inflamed and fills with immune cells? Like, what are some of the processes that contribute to this or fuel the inflammation in there? And so what we thought of at the time was the gut. After all, the gut is the first connection to the outside of the body for many of the things that from the environment, and that includes especially the diet. And so the diet and also the gut microbiota are basically foreign entities that are sitting across a single cell lining layer that the immune system has to kind of take into to consideration here. And so we were interested, like, how does the diet and the microbiome, connect with the, potentially the immune system living in the gut, and how does this all ultimately fuel inflammation in adipose tissue, which is a major feature, not just in obesity, but also in aging as well. Okay, so just a little bit of background. We're going to talk about the gut barrier and the intestinal immune system. And so the intestinal gut barrier is made up of a number of different components. There's basically a mucus layer in the small intestine, which is on the left. There's usually about a single layer of mucus, and it gets thicker to form a double layer of mucus as you go down towards the colon. This mucus is full of things that help keep the microbiome at bay. Some of these things would be antibodies. The most common antibody that would be secreted that kind of keeps the microbiome at bay and to maintain the barrier is IgA, so immunoglobulin A. It's a type of antibody. And you can see that it actually forms a dimer. There's like two of them in there linked. There's also these things called antimicrobial peptides. These are secreted by both the epithelial cells and also specialized cells called Paneth cells, and they also help keep the microbiome at bay, like preventing it from encroaching too far. The epithelials, the cells themselves, which line the intestine, also have their specific tight junctions and other junctions that help keep things out from coming into the body. And then some of these cells are ones like goblet cells. They make the mucus that I mentioned. And then you've got immune cells, which contribute to the gut barrier as well. There's a number of different type of immune cells, and they're present either in between the epithelial cells or just deeper down in the gut. In this area would be called the lamina propria, which is just underneath. All right. So what happens when you eat some food? So what happens is the food comes in, it's broken down into these little peptide chains, and which I call antigen. Antigen is anything that can be recognized by the immune system. You've got these macrophages, which express this marker called CX3CR1. They basically line the lining just under the gut epithelial wall. They capture these food particles and food antigens. They grab them and they engulf them, and then they actually load them onto these things called dendritic cells, another type of immune cell. These ones express CD103. They'll grab these dendritic cells will grab the food. They'll go down through the lymphatics, and basically they'll make their way to the lymph nodes. And so, mesenteric lymph nodes would be one example of draining lymph nodes from the intestine. There they talk to T cells inside the lymph nodes, and depending on the inflammatory environment, usually it's a anti-inflammatory environment in these mesenteric nodes, and that would activate the T cells in there to form regulatory T cells, which recognize these food antigens or can cross-react to them at least. And then these T cells then make their way back into circulation, ultimately, landing even back in the intestine, and that contributes to tolerance to the food that you eat. So the intestinal immune system, as I mentioned, has a very big role. It has a really big job. It's got to maintain tolerance to the food, but also to the microbiome. That's just, adjacent to it. The intestinal immune system, I forgot to mention, that's also the largest collection of immune cells in the body. As I showed you, it kind of, lines the inside of the intestine in multiple places. There's about 100 trillion microbes. There's more than 500 species of, bacteria with, even more strains from those. As you go down the intestine, there's more, amount of bacteria increases. And most gut bacteria are from, these two phyla, Bacteroidetes and Firmicutes. But there's some other types of ones that I might mention later in this family, in this phyla called proteobacteria, which is these ones are thought to be more opportunistic types of bacteria. Okay, so just like the adipose tissue, the immune system also changes during obesity, but also during aging and chronic diseases of aging. So in general, what happens is, if you're lean and healthy and you have a good diet, you have a, pretty good, functional barrier. These epithelial cells, which are the lining cells, they make, mostly anti-inflammatory cytokines. Another big one is TGF-beta. You've got, a pretty good amount of regulatory T cells in there making, anti-inflammatory cytokines. You've got a type of immune cell called innate lymphoid cells. These cells basically they look like T cells, but they don't have this antigen specific recognition. They basically make reparative cytokines, some of the ones that we're studying. And one of the main reparative cytokines in the gut is called IL-22, and we'll get into that a bit later. And so, there's not much inflammation in the gut when it's healthy. However, during chronic diseases, this could be during obesity, this could be inflammatory bowel disease, it could be a bacterial infection. You get a breakdown of the gut barrier. Bacteria is allowed, which are these rods and circles, they're allowed to enter, and then they can ligate these pattern recognition receptors. So these are the things that recognize bacteria. And basically, when you ligate or when you engage these pattern recognition receptors, especially on the penetrant or the lower half side of the intestinal epithelium, that signals an inflammatory response. And typically you get a shift in the type of cytokines that you make, and these epithelial cells and immune cells start making cytokines that are more typically found like in a bacterial infection. So the things like interleukin 1, IL-6, IL-12, IL-23, interferon gamma. And so you get this low grade level of inflammation or potentially a higher grade level of inflammation depending on the severity of the barrier breach. So we started looking at the gut and its immune system and insulin resistance back in 2011. At the time, there was hardly anything known about the gut immune system in obesity, insulin resistance, and actually, in terms of most chronic diseases, there was almost nothing known about what the gut immune system did, beyond inflammatory bowel disease. But in obesity, it was known at the time that obesity is associated with this unique profile of microbes. So and then the gut microbiome changes, and it changes, and when that term is called dysbiosis. And usually you get a reduced diversity in there and an enrichment in bacteria that can produce, potentially more, dangerous things to the host. It was also known that antibiotic treatment of high fat of rodents fed high fat diet can improve insulin sensitivity, especially when given later in the disease. suggesting that the microbiome could have a detrimental role in promoting insulin resistance. Now, this topic of giving antibiotics is a little bit tricky because the timing of when the antibiotics is given also matters. Like if you give antibiotics early during the court during like to a mouse or even to a chicken, it can actually worsen obesity. So it doesn't, necessarily always improve insulin sensitivity. And actually, for a while, I mean, if I'm not mistaken, people in industry would put antibiotics in chicken feed, for example, to make the chickens, gain weight more. Now, it was also known that was associated with this non-specific low-grade inflammation. There was a couple of papers just using very basic techniques across the entire intestine, showing that there's some increases in cytokines in there. And then there was also this classical paper showing that there's worsened permeability of the intestines, and you can get this leakage of bacterial products, mainly a bacterial toxin called lipopolysaccharide, can leak into circulation. So that was the state of the field when we had started working on this. And and here it is. This is basically kind of the state of the field. As I mentioned, this when someone who's lean and healthy, they have a really good diversity of the bacteria, and this is all strongly also shaped by the type of foods they eat, and we'll get into that. There's a lot of reparative signals and anti-inflammatory signals. During obesity, you get this reduced diversity of the gut microbiome. The gut barrier weakens. You get some seepage of bacterial products, including these endotoxins called lipopolysaccharide that leaks in, can activate the epithelial cells and the underlying immune cells to generate this low-grade, non-specific inflammation. But what really happens to the immune cells there? And that was kind of the project that we decided to do. And so we characterize how immune cells change in the intestines of initially of mice fed a high-fat diet, but some of this data has also been reproduced in human samples right now by other international groups. And so what we identified is that if you look at the immune cells inside a lean colon, and also people looked at this in the small intestine. As I mentioned, generally you've got, just like in adipose tissue, you've got this really homeostatic mix of anti-inflammatory immune cells like regulatory T cells, immune cells making reparative factors, so innate lymphoid cells. This is a type 3. There's a few types of these. The type 3 ones make this reparative cytokine called IL-22, which can act on the epithelial cells and cause epithelial cell and mucosal regeneration. It also, helps promote antimicrobial peptide release to keep the microbiome at bay. And also you make some of these again, like, antibodies that are associated with mucosal protection like IgA. So this is someone who's lean, someone who's eating a Western diet and obese. Basically, there's a shift in the type of immune cells. You get a reduction in these ILC3s that are making IL-22. So the gut barrier is not as maintained. You get a reduction in these regulatory T cells making IL-10. So, I mean, IL-10 can also, promote things like mucin production and help defend the barrier that way. And you get an influx in these other immune cells in response to this barrier breach, almost as if there's a bacterial infection, and they make things like interferon gamma. Um, and so this has been shown also in some human studies as well. But I mean, this is still, it's still an area of active research. Okay, so why is this important? Like, why is it important that the gut immune system becomes more inflammatory from eating a Western diet? And so it's important, from a mechanistic point of view, because when the gut immune system becomes more inflammatory, there will be, I break this down to three kind of, basic effects. There's effects on the gut barrier imparted by an inflammatory immune system. There's effects on how these immune cells traffic and what they do throughout the body. And there's also an effect on gut hormones. Okay, so the first thing is like during a Western diet, so this is a diet, high in, saturated fat, cholesterol, high in sugar, salt. So basically what happens is you get these inflammatory immune cells, and I mentioned they make cytokines, like a prominent one that they produce is interferon gamma. And so interferon gamma basically has been shown to directly break apart some of the junctions that are holding the gut barrier together. So when you've got these interferon gamma producing immune cells come in, it breaks apart the gut barrier. The interferon gamma also can change the gut microbiome and reduce some protective species in the microbiome too. But what happens is the interferon gamma made by these immune cells they break apart the gut barrier that allows easier seepage of bacterial toxins. These bacterial toxins, like lipopolysaccharide, get into circulation. I mean, they also get uptaken in part through chylomicrons, which is, where fat is uptaken. But the stuff that seeps into circulation can also make its way into fat. When it gets into fat, it acts as an adjuvant. So these bacterial products act as an adjuvant to the immune system and helps fuel inflammation in adipose tissue, for example. And the other thing that happens when the gut immune cells become more inflammatory, and this is, why it's relevant from a systemic point of view, is that these immune cells basically can traffic, to many different places in the body. So if you're having a Western diet, which is high in sugar and saturated fat, and your immune cells in the intestine become inflamed, what happens is these immune cells, they can traffic anywhere. I mean, so you can get inflammatory immune cells going to the brain. You can get them going to the joints where they may contribute to like arthritis. You can get them going to the liver where they contribute to metabolic associated liver diseases. And so because the gut is this focus, it's sort of like a central base of the immune system. And so when the gut immune cells aren't happy, it makes the rest of the body unhappy because they go everywhere. And so, just as an example, we just published a paper just recently showing that how the gut immune system in a mouse model of Alzheimer's disease can traffic throughout the body, and we were able to identify some types of B cells, which is another type of immune cell, inside the lining of the brain that recognizes commensal gut bacteria. So basically, if your immune system is recognizing, abnormal processes in the gut and it gets inflamed, this stuff is going to have systemic consequences throughout the body because this just by the way the cells are trafficked. Another reason why, having this, having an inflamed immune system in the gut can contribute to systemic processes is also its effect on gut hormones. So the gut is home to produce many hormones. Probably the most famous one now would be GLP-1. Now, when your gut barrier breaks down and your gut becomes inflamed, in the short time, basically what happens, you can get this acute sudden rise in GLP-1. However, in most chronic diseases, when this happens over a long period of time, there's been some studies that have shown that you actually reduce the amount of GLP-1 produced in terms of reducing the types of cells that can make GLP-1 because of the inflamed environment. And yeah, one of the things I want to mention, I'm going to be showing a lot of studies and papers from stuff that I didn't do just to try to keep it unbiased so you can see the breadth of the field. And so this would suggest that, having this, low grade level of inflammation in the intestine or this altered immunity also controls hormones. And here's an example where this has been associated with reduced GLP-1 production. And GLP-1, of course, is one of the main peptides that control insulin secretion, also controls satiety, and a number of different things. Okay, so that's why it's bad if the gut immune system becomes inflammatory in a Western diet or eating these ultra processed foods. And I mentioned because it controls the gut barrier that can lead to systemic issues. The immune cells can traffic to different places and cause inflammation, and they can also upset hormonal balances. But why does the gut immune system become inflammatory in the first place? And here I'm going to go through a number of the different pathways that contribute to the gut immune system becoming inflammatory, and some of the foods and the dietary patterns that contribute to this, and also some things that you can do in your diet to prevent this from happening and to keep your gut immune system, anti-inflammatory and happy. So the gut immune system basically becomes more inflammatory in a Western diet by eating ultra processed foods. It's mainly due to a combination of both dietary and gut microbial factors. Most of these gut microbial factors are things called gut metabolites, and so these are just metabolic products of the gut microbiome that act on your body's tissues. Okay. And so if you have like a lot of inflammatory, if you have a lot of inflammatory foods, just a sorry, that's just the excess of the virus. Um, so if you have a lot of Western diet or ultra processed foods, what happens is you get this the that acts on the gut microbiome, and the gut microbiome makes these metabolites, and you get this increase in these what I call pathogenic metabolites and a reduced level of protective metabolites. So let's talk about some of the factors here that are in Western diets that contribute to inflammation both in the gut and also in the body. And so, one of the first things would be if you have a diet which is high in saturated fat and cholesterol. So what does this do to the immune system living in the gut and also throughout your body? So for the longest time, it was thought that if you have saturated fats, these saturated fats would bind to the same pattern recognition receptors that recognize bacteria and then activate the immune system. I mean, that topic, while it may simulate that effect, the actual mechanism is a little bit more less understood. And so some people think right now it just has to do with these saturated fats getting incorporated into your immune cells and changing the lipidome, which is the cumulative amount of lipids in there, including the lipids that are in the membrane, and this alters the signaling properties of the immune cells. And by and large, they cause immune cells to make more inflammatory cytokines. So your macrophages would make cytokines like these would be chemokines, which bring in other immune cells. CCL2 would bring in like more monocytes. CXCL10 brings in inflammatory T cells. IL-6 and 8, these are IL-8 brings in neutrophils. IL-6 would be an inflammatory cytokine. And so this would promote inflammation. So it basically, reprograms the immune cells in the gut and throughout the body to increase inflammatory cytokine production. Another thing is sugar. So sugar is probably having a large amount of sugar in your diet is probably one of the worst things you can do to fuel inflammation. It's sugar, and this is basically how it works. So when you have a high amount of sugar in your body, it acts on both innate cells like these neutrophils, monocytes, and dendritic cells, and causes them to again make more inflammatory mediators. It also reduces the ability of your adaptive immune system to suppress inflammation, and so you get this inflammatory effect. And so how does this actually work? It works because sugar, when you flood your immune cells with sugar, it really fuels, glycolysis. So your metabolism can go through, a number of different pathways. Glycolysis is this rapid use of sugar, which your immune system likes to do to fuel inflammation. And it does this by a number of ways. I mean, I don't want to go into all of the details, but I thought this was a pretty good, summary diagram. It can do this by increasing the availability of calcium. It can do this by unlocking expression of inflammatory genes, because a lot of the enzymes used in glycolysis, or I mean, some of them can keep some of these inflammation related messenger RNAs at bay, but when glycolysis is actively engaged, it releases the inhibition on that, and you get all these new inflammatory messages going through. It can also, usually what happens when you actively undergo glycolysis, the citric acid cycle in the mitochondria basically has, a couple of sluggish areas and breaks. And so you get accumulation of byproducts from the citric acid cycle, including succinate and citrate, usually when you're revving up glycolysis like this. And basically, some of these products off the citric acid cycle can also, modify proteins and then also fuel inflammation. And here we have like interleukin 1 and more interferon gamma. So sugar is, probably one of the most reproducible things that you could do to fuel inflammation in the body. Another thing that's pretty common, so that's, high fat, high saturated fat, cholesterol, sugar, and here's another one, food additives. There's been a large increasing literature on food additives and what they do to the gut microbiome. Um, and so, uh, so maybe like some of the earliest ones that were identified would be like emulsifiers, and then later, artificial sweeteners. And basically, what these things do, so emulsifiers would be things in the diet like carboxymethylcellulose, polysorbate 80. So when I'm looking at foods, like I always look to see if there's emulsifiers in there. I try to reduce that in my diet as much as I can because there's been a number of studies right now where if you eat these things, emulsifiers, they're like thickening agents in the food and they help prevent separation of like oil and water phases. Um, However, they have, effects on the gut microbiome. Some of them are metabolized, most of them are not, though. And what they do is they can modify the types of genes that are produced by the microbiome. And one example is they can, increase the amounts of these of lipopolysaccharide. They can increase expression of flagellin, which is another pattern that's often recognized by the immune system. So LPS is recognized by this molecule called toll-like receptor 4. Flagellin is recognized by this thing called toll-like receptor 5. And so these emulsifiers can increase the pathogenicity of the microbiome and reduce their diversity. And it's been shown in a couple of studies now that by reducing the gut barrier, and you can activate inflammatory pathways and contribute to low-grade chronic inflammation that I described with obesity and insulin resistance. And as you know, when your gut immune system becomes inflamed, as I mentioned, it predisposes the rest of the body to the same effect. Now, artificial sweeteners, these would be things like probably the best studied would be sucralose, saccharin, aspartame. Um, Probably sucralose and saccharin, I'm going to say, are the two ones, if I had to pick two, that are most, associated with this effect. But I think they've all been shown in some way or another to have some effect. And one thing I want to mention is that, it's not everybody responds evenly to these things. So it's not like if you give like, emulsifiers to 100 people or saccharin or sucralose to 100 people, you're not going to get 100 people with an inflamed gut. It's also the microbiome of each individual people, which will dictate. Some people have a more vulnerable, personalized microbiome that are more susceptible to these things. But when you give them in mice, they tend to work, in almost all mice. Um, Now, so these artificial sweeteners, these ones again, like these ones to me are a little bit less clear how, they have been shown to reduce bacterial diversity. And they're a little bit less clear. They're generally not metabolized. We're talking about sucralose here, and they're not metabolized by the bacteria, but it may be just that they interact with it and modulate gene expression. But I'm not, completely sure of the mechanism of that. But it has been shown that, it reduces diversity of the microbiome, and you get enriched for bacteria that maybe has, more tendency to produce, lipopolysaccharide, reduced capacity to make beneficial metabolites such as short chain fatty acids like butyrate. And again, a similar story to the emulsifiers. They can act on these epithelial cells in the gut, signal inflammatory cascades, and then contribute to low-grade inflammation. Food colors, this is something I look at. I don't give my daughter food colors if I can. This is one thing I try to avoid like the plague. Um, So food colors, I mean, and the reason I do this is because these are completely unnecessary. I mean, some of these other things, so sweeteners is tough, right? Because I mean, glucose is really bad also. I mean, and so if you want to get the taste in there, it's like you can't give too much sugar because it's bad, but the sweeteners aren't so great either. So that's a much more difficult game. But food colors, I mean, I don't even know why these are needed at all. There's definitely natural alternatives to this. So, I had some ginger in my sushi the other day. It was red. I'm like, holy crap, I'm eating red 40 right now, right? So that that one, yeah, there's no reason at all to my knowledge to use food colors for anything at this point. So I mean that might be, a little bit polarizing and a strong statement. I realize it, but I mean the literature is starting to support, that these things can also modify the gut microbiome. So for example, red 40 and yellow 6, these have azo bonds. They're relatively stable. They're not digested so well by the host, and so they make their way to the microbiome. The microbiome can actually metabolize these guys and form these compounds called ANSA. I think are these sulfonic acid compounds, if I remember. And so basically, these things can then act on the immune system and basically promote inflammation. Now, just like sweeteners and emulsifiers, it's not everybody. You need to have the right environment basically for these food colors to, promote inflammation in there. So it's not like if you give red 40 to everybody, every human is going to basically start making IL-23. IL-23 is a cytokine that, causes a number of different inflammatory cascades and T cells. But basically, it has been shown, at least in, mouse models, and it's the data starting to come out now in humans that, like these things across the board, emulsifiers, sweeteners, food colors, can contribute to inflammation in the intestine. The last thing I mentioned, just nanoparticles. So these are things like, I mean, the classic one would be titanium dioxide. I had this one yesterday in a little cookie I had. I mean, it had a yogurt coating which had titanium dioxide in there. And I read these labels now and I'm like, wow, like everything I eat has this kind of stuff in there. And so I try to reduce it as much as I can. But titanium dioxide, again, like, kind of helps, hold the food together. Also been shown to reduce bacterial diversity, reduces some beneficial microbes, including ones that make short chain fatty acids. I'll get into that a bit later. And again, these things can also activate inflammatory pathways. This one activates the NLRP inflammasome, which is the main thing that produces IL-1 beta, a main inflammatory cytokine implicated in many different chronic diseases. Okay, red meat, something else to try to avoid, and not to eat every day. So, I mean, we've talked about saturated fats, cholesterol. We've talked about, food additives and sugar. Now let's talk a little bit about red meat and what this does. So, red meat so we're talking a little bit about this thing called trimethylamine N oxide, TMAO. TMAO is naturally occurring in some foods. I mean, interestingly, fish has a lot of natural TMAO in, but red meat, for example, has a lot of the precursors for this. And so red meat would have something called, carnitine in there, and also and there's also phosphatidylcholine and choline in a lot of this animal types of foods. And so when you eat this red meat, this carnitine gets broken down eventually into the liver by something called TMA. So I'm sorry, it gets broken down, by the gut microbiome into TMA, and then eventually in the liver it's converted to trimethylamine N oxide. So TMAO is the one that has been linked, where its levels have been linked to cardiovascular disease and a number of other issues. But what it does to the gut is also interesting. It again, uh, when this stuff gets into the gut, it promotes inflammation, and it does that by acting on three major pathways. One, it can induce NF-kappa B, which is a major integrator of immune inflammatory signaling. It can activate this protein called PERK, which is part of the ER stress unfolded protein response. They can also activate the inflammasome NLRP3 I mentioned, which contributes to IL-1 and IL-18 production. These are all inflammatory cytokines. But one of the more scarier things these TMAOs can do, especially ones that are inside the GI tract, is that it forms these N-nitroso compounds like nitrosamine and things like that, and these are carcinogens, and so and these carcinogens can damage DNA. They can also contribute to oxidative stress and even potentially contribute to colon cancer formation. And so red meat. This is why people generally recommend not to have this, every day because it contributes to things that are linked to cardiovascular disease and potentially uh, cancer and inflammation. Another type of thing. This one is interesting as well. So TMAO, and then other types of things that can act and become inflammatory on your immune cells would be these indoxyl and p-cresyl sulfates. So these things are known to go up with aging and also chronic diseases of aging, and they contribute to, again, cardiovascular disease, and they're also not very good on kidneys. So these are started by taking, and again, if you eat, this is like eating too much meat or too much protein, for example, things that are rich in tyrosine and tryptophan. Now, this is interesting the way indole is formed because tryptophan metabolism produces a lot of really healthy metabolites. But indole is one which is actually healthy, but it's not. But when it gets converted to by the liver to indoxyl sulfate, then it becomes unhealthy. Now, and I'll get into tryptophan metabolism in a bit, but basically what happens is tyrosine and tryptophan are converted by the gut microbiome into compounds called p-cresyl and indole. There should be an E at the end here, and then as they make their way to the liver, they form p-cresyl sulfate and indoxyl sulfate. These things are also known as uremic toxins. So they're called uremic toxins because, I mean, they're not very good on the kidney. They cause reactive oxygen damage in the kidney, but they're also not very well cleared by dialysis. And so if someone has chronic kidney disease and they're on dialysis, the level of these compounds go up and they're not as cleared effectively through dialysis. If you're young and healthy, you can generally clear these reasonably well. Now, so where are these? Again, these are in like the meats. P-cresyl is also directly present in smoked foods. So things like sausage, salami, like these ultra processed foods, you get like, a lot of this, a double whammy of having like the red meat, but you also get, you get like the TMAO. I wanted to mention something else about TMAO. I'll do that after I go through here. Um, but basically, these are present in these things. They form uremic toxins. Uremic toxins can act on a number of different immune cells, but probably the thing that's been best studied about uremic toxins is they act on the lining cells of the blood vessels. And so these would be endothelial cells, and they can crank up expression of a whole bunch of these molecules that allow immune cells to migrate in. They can contribute to reactive oxygen species production in these cells and overall increased levels of inflammation across the vasculature. Now, I wanted to mention just one quick thing here about these N-nitroso compounds as well. And basically, this reminds me of another type of food that I probably should have put in here that I forgot. And this is another preservative that's commonly used in ultra-processed foods called sodium nitrate. So nitrate and nitrite. So sodium nitrite would be something that's in a lot of, again, like sausages, hot dogs, deli meats. And then what happens with these things is this a very similar thing to TMAO. What happens is it's the acid in the stomach and the meat proteins in there act on these nitrites and they form these N-nitroso compounds like nitrosamine, and they're carcinogens basically. So I mean, just know when you're eating like a hot dog with sodium nitrite in there, you're eating something that, has a pretty good evidence. I mean, the nitrite is not a carcinogen, but the byproducts of that are pretty high up on the cancer causing scale. I think it's a 2A or something. Like it's likely, but it's there's a lot of studies on that. And so yeah, so when you're eating like the red meat, so if you're eating like you're getting the TMAO forming the nitroso compounds. If it's got sodium nitrite in there, you're getting the sodium nitrite forming these carcinogens. If it may have P. cresol in there also, so you're getting the ability to make uremic toxins if you're having sausages. And so and then if you barbecue it, you're getting all these other hydrocarbon cancer causing stuff forms. And there's no, I mean, you can imagine if you have a diet of like, like a barbecue hot dog and like a burger and fries and processed and bacon in the morning, and you have this, for a long periods of time, it's no wonder. And then you wash it down with like a, high sugar beverage. It's no wonder your gut is inflamed and people are starting to get colon cancer in their 30s and 40s. It's, the diet is a major thing here that's contributing to it. But again, there's also a personalized component to this with the gut microbiome. Okay, so that's the doom and gloom. Let's look at some of the things that you could do to to protect yourself from this. So people ask me, what are some of the best things you can take for your gut immune system? And probably the ones I would recommend first, I mean, have more dietary fiber. So dietary fiber is very protective for the immune system in your gut. And what it does is dietary fiber acts on the microbiome. Some of the types of bacteria in there are known to be fermenters of soluble fiber, and they form these things called short chain fatty acids. These short chain fatty acids then make their way into the immune cells, and they are known to directly unlock gene transcription, especially in regulatory T cells, and boost, anti-inflammatory cytokine production through a couple of different pathways in here. So dietary fibers have been shown to basically dampen inflammation, and they do it by boosting regulatory T cell function and other types of immune cells here as I list. Another thing that's good would be vitamin D. Most studies on vitamin D usually do about 2,000 units per day. Oh, and by the way, these are, I'm not giving like, you always consult with your physician before, changing your diet and whatnot. I mean, I am a medical doctor by training, but this is not designed to tell you what to do. I'm just providing you the scientific literature that's out there. But vitamin D, basically, again, has been shown, in most studies that mention about 2,000 units a day, has been shown to boost regulatory T cell function. So a lot of immune cells have the vitamin D receptor in them. Vitamin D will make its way in there, dock onto the receptor where it modulates thousands of genes. And generally what they do is it dampens inflammation. Again, it produces this cytokine called interleukin 10, and that has the ability to dampen the inflammatory immune cells in the intestine. Omega-3 fatty acids, this would be another good one. Most studies generally do about a gram a day of combined omega-3 fatty acids. And basically omega-3 fatty acids, they incorporate into the cell, they get metabolized, they form these things called resolvins. That's what these RVs are. And these resolvins can dampen inflammation. That reminds me also, like, so these are omega-3 fatty acids. It's important to have a decent ratio of omega-3 to omega-6 fatty acids. So another type of polyunsaturated fats are omega-6 fatty acids. The literature is a little bit more varied on those. It used to be that if you had a lot of these omega-6 fatty acids are found things in vegetable oil, right? Like sunflower oil and things like that. And it used to be the thought that if you had a lot of these omega-6, this was the culprit causing inflammation because these omega-6 fatty acids would basically, integrate into the cell membrane and contribute to inflammation by cyclooxygenase and lipooxygenase, which are enzymes that break down arachidonic acid. But the data on that has been, at least in the human trials, has been a little more ambiguous whether omega-6 fatty acids are truly responsible for driving inflammation. But what has been reproducible is that omega-3 fatty acids do shut down inflammation in many trials. Um, and they do this by, they induce these resolvins. These things inhibit NF-kappa B, which, as I mentioned, is a major driver of inflammation in the body. It's a major factor that induces transcription of many inflammatory genes. It also can activate PPAR gamma, which also has reparative effects and anti-inflammatory effects. It can also, these resolvins can also, directly work on immune cells, and these would be the macrophages, reducing things like IL-1, 6, and TNF, and boosting these inflammatory cytokines, these anti-inflammatory cytokines. And if you remember, these are the same type of ones that you found like in the healthy adipose tissue, and also to a lesser extent, like the healthy intestine. And so, I mean, omega-3 fatty acids, I mean, there's been some studies. I mean, there has been some ambiguity with whether they reduce, mortality, but there has been some studies showing that if you combine omega-3 with vitamin D and the right amount of exercise, you can potentially slow your rate of aging. And I think it's over a 3-year study, if I'm not mistaken. I mean, you might know these better than me. I believe it was about 3 months at least of slowing aging on DNA methylation clocks for every 3 years, if I'm not mistaken. And so these, I mean, which is quite remarkable still from inducing relatively simple dietary and lifestyle interventions. AHR metabolites. This is something I wanted to talk a little bit about. Tryptophan metabolites. So this is something else that I would consider would be pretty healthy. So cruciferous vegetables. Broccoli is a source of AHR metabolites. So it has this thing called indole 3 carbinol in there. It's broken down by the acid in your stomach. It forms these things called AHR ligands. These ones generally bind very weak to this aryl hydrocarbon receptor, which is present in a number of immune cells. It's in these immune cells that are wedged between the epithelial cells. It's also in some of the innate lymphoid cells and some of the regulatory cells. And then this docks on into the cell and instructs it to make a number of different reparative cytokines, including interleukin 22. So broccoli is good. So tryptophan metabolism. Most of the tryptophan that you eat is metabolized by these enzymes by the host. So you would be called TDO and IDO. About 5% or so, or less, makes its way to the gut microbiome, where it's metabolized by different types of bacteria. So if it's metabolized by this one called Clostridium sporyngenes, it forms this molecule called indole propionic acid. If it's metabolized by different types of lactobacillus, you get indole 3 aldehyde. And these things are highly protective to the gut immune system. Indole propionic acid has basically antioxidant and neuroprotectant effects. It binds to this thing called the pregnane X receptor, but this one also binds to the AHR, the aryl hydrocarbon receptor, to a weak amount. And when you ligate the AHR, weakly in these immune cells, you produce, good amounts of this cytokine called IL-22, and IL-22 directly programs the gut barrier to promote repair. As I mentioned, it also can induce antimicrobial peptides from some other of these cells in there to keep the gut microbiome at bay. Now, indole, which of course is the precursor for indoxyl sulfate, and this is where it gets a little bit interesting because indole basically is made by tryptophanase expressing bacteria. So generally, these are produced more in people who have a pre-established dysbiotic microbiome. So if you've got like E. coli floating around, a lot of E. coli or Klebsiella or types of Proteus, I can't remember some of the other ones, but these are ones that have higher levels of tryptophanase expressing bacteria. So if you keep your gut microbiome healthy, a lot of this tryptophan is metabolized into these ones like indole 3 aldehyde, indole propionic acid. And you'll get some indole, but smaller amounts of it. Indole on its own is actually really healthy. It's been shown to promote things like GLP-1 and improve tight junctions. And if you're healthy, producing indole and getting a little bit of indoxyl sulfate gets cleared really, really well by the kidney. However, it's just when you get older and your kidneys start failing, or if you then at that point, this indoxyl sulfate can accumulate as a uremic toxin and cause vascular inflammation. But tryptophan byproducts are generally really healthy, um, and they're reparative and anti-inflammatory on the immune system. These indole metabolites. Most of the tryptophan though that's formed in the IDO TDO pathways or the kynurenine pathways, a lot of those lead to inflammatory metabolites though, which we're not going to get into today. And so, yeah, as I mentioned, if you look across chronic diseases of aging, these indole metabolites or these AHR ligands have been shown to go down. This has been shown in both mouse and a number of human studies right now. There's a lot of human studies that have measured the levels of some of these indole metabolites, and they basically go down during many chronic diseases of aging, including type 2 diabetes. And the reason is they have a remarkable effect at improving the gut barrier because they make things like IL-22. They can also promote GLP-1 production. Okay, so basically, um, this is so I'm trying to put this all together right now into a summary figure over here. So how does the diet that you eat and the microbial factors that are produced, how does this control gut immune function? What are some things that you can do? So if someone's eating a really good balanced diet, so I mentioned, so dietary fibers, really important. These form short chain fatty acids, which help, increase, Treg function to produce, IL-10 and TGF beta. These cells, I mean, these factors and these cytokines dampen inflammation in the gut, but they can also promote mucus production. They can also promote IgA antibody production, and that keeps, the gut barrier intact. Another thing, eating a lot of cruciferous vegetables like broccoli, which has these precursors for AHR ligands, that activates immune cells with the AHR receptor, and these make things like IL-22, also lower levels of some IL-17, and these, cause antimicrobial peptides produced. So you can imagine, so eating fiber and, like cruciferous vegetables, I mean, you get a pretty good amount of, gut barrier protection right there. Eating things like omega-3 fatty acids also directly dampens inflammation. So these are things that you can do to protect the immune system in your gut, keep it less inflamed. And as people get older and they start eating, Western diet, a number of things happen. Often people don't get enough vitamin D, so that predisposes your immune cells to become more inflammatory. They don't get enough omega-3 fatty acids. They eat a number of these, ultra processed foods. We just put in one example here of like, TMA, which can form TMAO. And then you get this, chronic inflammatory environment in the intestine. You see this with chronic diseases of aging. You get, weakened gut barrier. You get seepage of microbial products that fuel inflammation in the body. And so basically, this contributes to intestinal immune dysfunction, which has systemic effects across the body. So when I started my career as an obesity diabetes researcher, I always thought the adipose tissue was the center of the universe in metabolic diseases. But I kind of reshaping this model to think that the gut and the gut immune system and its interface with the microbiome now is the center of the universe in chronic metabolic diseases. And so I mean, so this is how a model that we think works for obesity, insulin resistance, and also many chronic diseases would probably follow something similar to this. And so it's basically changes to the diet, alters the gut microbiome, changes the metabolites they produce. These changes in metabolites alters the immune cells living in the intestine, but also alters the intestinal barrier itself. This causes more gut permeability, which allows seepage of bacterial toxins. Some of these get into adipose tissue where they got more bacterial toxins acts as an adjuvant, which fuels inflammation and fat. This inflammation and fat would cause insulin resistance in the adipose tissue. Insulin resistance in the adipose tissue then causes production of cytokines that go systemically, but also free fatty acids that can get systemically, they can get themselves into the muscle, they can go into the liver. When they get into the liver, it can help promote things like fatty liver disease and also metabolic associated steatohepatitis so inflammation in the liver. These immune cells can also traffic, as they get inflamed, can traffic, to the brain, to the joints, to the liver, to multiple different tissues, and contribute to inflammation and low-grade, deterioration that you see with age across many different diseases as well. Having this, abnormal, gut inflammatory changes in the gut can also potentially alter gut hormone production. And one of them I mentioned was potentially reducing GLP-1, which can then, feed back and just worsen all of these conditions. And GLP-1 itself has some anti-inflammatory effects on immune cells. It can do it, through talking to immune cells, but also through brain circuits, through brain immune interface circuits was what recent papers have shown. And so basically the gut immune system, I think is a center point for not just, obesity and chronic metabolic diseases, but most chronic diseases of aging. And that's just emerging literature that's coming out. And this is all shaped by what you eat, really. Like it's one of the main things, how the diet, goes to the microbiome, which then goes to the metabolites, which then shape the immune cells, as I've illustrated. So in conclusion, these are some of the conclusions we have here. So the adaptive immune system, and also the immune system in the gut in general, it's just an important participation in the regulation of blood sugar. The gut immune system has control on the gut barrier, but also these immune cells can traffic throughout the body and they can also impact gut hormones. Um, Diet and gut microbial factors are two of the main things that influence the level of inflammation in your gut immune system. And then the gut immune system controls systemic inflammation. And I didn't even get into some of the other ways that this could happen also. A lot of the things that the bacteria, if your gut immune system can recognize bacteria, there's also a lot of cross reactivity that may happen on your self antigens. And so, recognizing, some bacteria in your gut could potentially predisposed also to autoimmune disease by cross reactivity onto your other tissue. And we didn't get into that. And then when you're shopping for your food, I mean it's okay to have a little bit of this, but if you have this a lot, you're going to go down a steep road of having health issues, and especially look out for saturated fat, trans fat, too much cholesterol. So food additives is a big one, like some of the emulsifiers I listed, some of the sweeteners I listed, some of the nanoparticles I listed. Um, so food additives, red meat, don't have it like every day. Smoked foods, especially, don't eat those every day. These things stress the gut barrier and they can trigger gut inflammation. Some anti things that you could do to protect your gut barrier and your gut immune system. So again, like, don't go overboard on taking vitamin D, for example, because there's also been studies that if you have too much vitamin D, like mega dosing vitamin D, you can eventually also worsen your gut barrier. I think it was one study I saw, like megadose vitamin D. Oh man, I don't remember how many units it is, but it's like over 10,000 units at least. Like don't megadose vitamin D unless prescribed by your doctor, of course. But things you can do like in the right amounts. So dietary fiber, vitamin D, omega-3 fatty acids, these things help, promote a healthy microbiome. Cruciferous vegetables that give you indole metabolites, which are nice. Um, these things dampen inflammation and then promote gut happiness. And I mentioned the company, one of the companies I founded, Propion. We work in indole metabolites, and we have a shake that can, gently, raise some of the beneficial indole metabolite levels as well. But overall, the gut immune system is a new major regulator of chronic disease of aging. So this would be insulin resistance. There's also papers showing that this contributes now to neurodegenerative diseases and and other diseases. And I want to thank all the people who helped. I hope I didn't miss anyone here. It's been a long road with, I've worked at a couple of places, mostly at UHN and also the Buck Institute for Research on Aging would be the two main places I've worked at in my career. And just want to thank the people and some of the collaborators. And a take-home message, I mean, don't forget to eat your broccoli. I mean, it's got a lot of effects. You've got the fiber in there, you've got the AHR ligands, you've got the sulfonamine, which I didn't even mention. I didn't talk about polyphenols and some other things. Don't forget to eat your broccoli. It's good. I mean, that's one thing I try to give my daughter. I know kids don't like eating vegetables, but at least try to get them to eat a little bit of broccoli as much as you can. All right, and that's what I got for today. It's a kind of a big walkthrough of pretty much the whole field almost as much as I could. I didn't get into bile acids either. There's a lot on bile acids too. It was just getting long, and so I thought that was probably sufficient for now.
That was great. I love deep dives. My viewers love deep dives, so that was fantastic. Gut immune system, gut metabolites, all of that. So I just want to add a little bit of context. So I think the high fat, high sugar Western diet, and then the HFHS model that's used in animal models, it's a bit of an over-representation of the adverse metabolic health that results, because the fiber source that's used in the animal studies is cellulose, which is not fermentable. So basically, the high fat, high sugar diet in animal models is a model in part of what happens if you don't eat any soluble fiber, right? And then you can kind of make the corollary in people who are eating just an ultra processed food diet, which is going to be high fat, high sugar. There too, they're not eating any soluble fiber. So some of the effects, I think, of the high fat, high sugar diets, both in people and in animal models, are mostly a soluble fiber story, because, as you know, Patrice Kenny's work, when they add soluble fiber back into the diet on the HFHS animal models, I mean, improved insulin sensitivity, reduced body weight, so many positive metabolic effects. So what are your thoughts on soluble fiber being an important part of this story?
Yeah, I mean, soluble fiber. Yeah, so I mean, right now they do have like if I mean, we get a lot of our stuff from you can get them from Taconic or Jackson Labs or Research Diets. We get our diets though from Research Diets, which is one of the companies, and you can buy controlled diets too, which match fiber. However, I agree, if you're using like a study in mice and you've got your normal chow diet, from whichever facility you're working in and you're comparing that to a high fat diet or high fat, high sugar diet, those two aren't going to be matched properly for some confounding issues. And one of them would be fiber content. Now, I agree. I mean, we've actually looked at the effects of cellulose and also versus, and there has been studies that have done this too, looking at it like inulin versus cellulose. And so we've done these kind of studies even in our mice with Alzheimer's disease. And so cellulose is basically, as you mentioned, it's non-fermentable. It's kind of like roughage. It passes like right through kind of. It does add some bulk to the stool. But inulin is or another fermentable or soluble fiber would be like something that the body, the gut microbiome can metabolize, form these short chain fatty acids, and then that can have effects on immune cells. And so basically, yeah, so we see like, bigger effects. Generally, when we do studies, we see bigger effects with inulin than we do with cellulose for those immunological reasons. So yeah, usually when you do studies, I mean, recommend that you basically get the matched control diet when you can. That this is matched for a number of different macro and micronutrients and things like fiber as well. One of the things that they add to these diets, though, is they add like they do add food coloring to the diet, so because you want to label, which diet is which, and so your Western diet could be green and your control diet could be yellow, but they both have food colors in them, and so that's another kind of confounding thing with diets, which I mean they do it. I mean, I didn't that would be one reason, I guess, that from a research point of view, why you might need food colors. You don't mix up the diets. But yeah, no, I mean the fiber would have a profound effect. And it does so because it also expands, like, good types of bacteria that can, metabolize fibers. And some of those would be like Fecalibacterium prausnitzii, like the Akkermansia, some of the bifidobacterias. And in our Alzheimer's study, I mean, bifidobacteria was expanded like, massively by giving inulin. And bifidobacteria is a good one. I think that's found in a lot of yogurts, can make short chain fatty acids, improve the gut barrier, but it also produces other metabolites I didn't even talk about today, some of the polyamines. So polyamines are produced by bifidobacteria, I think some of the lactobacilli as well. And these are things like putrescine, spermidine, spermine. They basically also dampen inflammation. And I mean, these are other factors that are in there. And so, this inulin basically, or why fiber is so important, it expands these good types of bacteria also, in addition to having them produce short chain fatty acids, but it also gets your gut microbiome in a healthier format. And so imagine if you're, having like some, more tryptophan, and you've got a healthy microbiome, you'll be making more things like indole acid, indole aldehyde. You'll still make a little bit of indole, but you won't make it to as much of an extent as if you've got a dysbiotic microbiome. And so, yeah, I agree. I mean, soluble fiber, it's one of my top three or four things to take. And I mean, one of the other things I forgot to mention would be another thing I would recommend for people is to take like a multivitamin, just in case you're deficient in something. Bacteria can also produce some of the B vitamins, but like some people are deficient in some vitamins. So, I mean, for me, like it'd be like a multivitamin. Some of the other ones I mentioned, soluble fiber, like fiber for sure is top four, top five for me in terms of how that's something you could do to protect your gut barrier and gut immune system.
The other thing that produces short, or at least increases short chain fatty acids, is exercise training. So, so the mice that are on a high fat, high sugar diet, they're sedentary with no soluble fiber, no exercise. This is like a
Yeah.
nuclear bomb of terrible health. So, all right, wait. So just digging deeper into mechanisms. So for people that are eating a high fiber, whole food based diet, you should have an increase in short chain fatty acids in the proximal slash distal colon. That's a whole nother question that I'll have in a minute. But so the short chain fatty acids, as you know, acidify the colon, right? Which then should limit the growth of things like proteobacteria, as colonic acidification limits the growth of proteobacteria, which can produce some of these metabolites like indoxyl or pericresol or TMA. So I think that too is an underrated aspect of quote unquote gut health, where colonic acidification, whether that's eating a high fiber diet combined with exercise, but then it even raises a question of there may be specific foods based on, a person's gut microbiome for personalization, where even on a whole food based diet, one can have higher than expected levels of indoxyl or pericresol or TMA because they're not eating the right fibers for their microbiome. So what are your thoughts on colonic acidification and the personalization, of fiber towards the microbiome?
Yeah, so for me, it's not just the acidification. I mean, a lot of the metabolites that are produced by the microbiome, whether it's lactic acid or some of the tryptophan metabolites, some of the indoles, these are known to actually feed back and control levels of other types of microbiomes, of other types of species in there. So every time a bacteria produces a metabolite, there's going to be an effect on the niche in there. And a couple of them that have been studied. I mean, my lab has looked at some of the indoles. I mean, some of the indole metabolites basically feed back and can act almost as low levels of antibiotic kind of effects. It can dampen the proliferation of some species. In fact, actually, interestingly, some of the indoles, for example, can produce can reduce mycobacterial levels, which is quite interesting because I know people have been looking for new therapies for tuberculosis and other mycobacteria cause species, and a lot of them are probably right under our noses in the gut. I mean, if you can study some of the metabolites and what these things can reduce, that these are potential uses. Now, in terms of lactate or acidification in there, I believe there has been some studies that have shown that like lactic acid, and I'm going to have to look this up, can also directly contribute to worsening blood sugar as well. Um, and I'm going to have to look that one up. But I agree. It's like if you're controlling, the acid environment. Now, for me, like the acid environment can keep some species at bay, and this is actually a good point because this is another way that people can potentially, if they're going to process food, like a deli meat, for example, instead of putting the sodium nitrite in there, I believe you can also have some sort of, acid treatment to replace that. Maybe give it acid with a certain amount of pressure can actually reduce the need to add some of the preservatives. I mean, the preservatives like sodium nitrite is added specifically. I mean, it contributes to the flavor, but it's also in there to reduce some of the species in there, like some of the Clostridium in there, if I'm not mistaken. And so, you can get rid of some of those through some of the acidification things in there, which probably is analogous to what's being described in the intestine to control some species that don't thrive as well from the short chain fatty acids. But I mean, short chain fatty acids, to be honest, I mean, they contribute to that, but I think they're also fuel sources for your endothelial, your epithelial cells in the gut. And so I'm an immunologist, so I mean that's probably the main thing I study is that how their benefit to the gut immune system, but also to some of the to keeping the gut barrier intact, also by acting as a major fuel source for the lining of the gut as well. Um,
Uh,
but yeah, no, I mean, in terms of, like what is like a more acid environment, could that do to the immune system, for example? Um, I have to look at some of the acids that are produced, as byproducts of metabolism in the immune system. I mean, we don't actively study this, but as I mentioned, like lactic acid, as a byproduct of metabolism is one of the things that I know has been looked at. And I believe there's been studies in diabetes that have shown that has linked this actually to higher glucose, actually.
Yeah. So the challenge, though, is if short chain fatty acids are important for limiting the growth of bacteria, pathobionts like proteobacteria. If you eat a high fiber diet and don't get a boost in short chain fatty acids, it's like, what do you do? So, as an example, we finished a clinical study at Tufts where we fed people, older adults, older than 65, a whole food, high soluble fiber based diet. So, the high fiber group ate about 60 grams of fiber per day, 2x more than the control group. And we saw very marginal increases in butyrate and propionate and no change in acetate in fecal levels. I mean, it was like 18 millimolar versus 14 millimolar for butyrate. I mean, very small but significant increases with a 2x increase for soluble fiber intake and no change in plasma levels of short chain fatty acids. So optimizing quote unquote gut health through short chain fatty acids doesn't seem to be as obvious as just eat a crazy amount of fiber and you'll get an optimal amount of short chain fatty acids in the gut. So I don't know, is this a polyphenol story or you got to add other components? How would you further optimize gut health if it can't just be with eating more fiber?
Yeah, I said it's got to be like a kind of a holistic approach. And I mean, how long was that study? Because my feeling, so I mean, usually when you so the gut microbiome can change within 24 hours, but it's more controversial for extended changes in the microbiome. Um, but the studies that in terms of like you know, microbiome, as you said, just like and there's other controversies too. Like just like if you have a high fat diet, and then you take and the only change you do is you add inulin to your high fat diet, right? So I've seen studies. I mean, these ones were in mice though, where if you continue your high fat diet or your Western diet and only you've added an inulin, and now you're also getting predisposed to liver cancer. So there was a study showing that yeah, so just adding in one variable isn't always going to be, the thing that you can do to correct it. And again, it's a holistic approach. I mean, you need to, I mean, combine, I mean, the diet is probably the most important thing that contributes to your microbiome, but you have to do this also, as you mentioned, in the context of proper exercise, proper sleep. Sleep, I mean, the circadian, rhythm controls the immune system. If that's off, and your immune system becomes inflamed. So say you're not sleeping, then your immune system becomes inflamed, and then the cytokines that you produce exert pressure on the microbiome, and that can also, cause dysbiosis in there. And so there's so many factors. I mean, it could be what if the people who got the short chain fatty acids weren't sleeping so well, then they're not going to produce they're not going to who that got the soluble fiber. They're not going to be producing optimal levels of short chain fatty acids. But at the same time, I mean, so they will produce, and then the question is, which I don't know is, how much of an increase do you need to exert a biological effect, right? I mean, you may only need, a very small increase to exert, dramatic biological effects. Like I don't know what the thresholds are. But that reminds me though, like of the whole NAD story too. So I mean, the levels in the tissue may ultimately, be more important than plasma levels. So I mean, there was a big NAD story the other day showing that people, as you get older, it was always thought that NAD levels were gonna go down in circulation, but I think the study showed there was no change in NAD in plasma levels. But we don't know what happens in the tissue, and then it could just be that small changes in the tissue get amplified, for their physiological effects on immune cells, and other cells. But as I mentioned, I think it would be more of a holistic approach, multiple changes to your diet, rather than just adding in one item and then combining that with some of the other environmental effects, as you mentioned, exercise, and then also, sleep habits. And, when you combine everything, I would imagine you would get, probably more, significant or noticeable effects. I mean, when we did do it with our mice, though, this shows you the difference between mice and humans. When we added inulin diet to our mice, we get, massive increases in short chain fatty acids, like not tiny amounts. We would get, you know, like two, three. I have to go back to some of the papers we did, but it's fold changes, right? Like twofold, like threefold, right? Not like incremental, small but significant effects that you're describing. And so that also goes to show you that, the comparing, stuff translating from humans and mice that don't always line up. In humans, it's because there's, as I mentioned, a lot of other things going on. I mean, there's, sleep, diet, exercise, and also like the existing microbiome. When we give it to mice, basically, like they all start off, they're litter mates, so they start off with similar microbiomes, but every human has a completely different microbiome. And as you said, there's other factors in there. Like if you're trying to expand, if someone's got dysbiosis already and you're trying to expand like short chain fatty acid producers, it may take, a longer extended period of time to get effects on those if they're starting off, at a disadvantage. But the other thing is, I mean, it would be interesting to look at if there was a microbiome signature for the people who responded better. Is probably what I would do on that study to see, if people who responded better with higher levels of short chain fatty acids, do they have a different microbiome to start? Because as I mentioned, with many of the food additives and everything is moving more towards a personalized nutrition approach where you need to have a certain microbiome to start, which will dictate, how you respond. And I think some of those studies, even I think the classic one was the one I remember on bread that was done in Israel, I think, by that team. And not everybody would respond the same to bread. And I think they did the same thing with saccharin, like artificial sweeteners, if I remember. And yeah, so each person will respond differently to a dietary intervention based on their existing microbiome, and that's another kind of confounder I'd probably look at in that study.
Yeah, so it was a 12-week study, and we didn't give any fiber supplements. We didn't give short chain fatty acids as a supplement or inulin as a supplement. It was a whole food based diet. So basically, when you talk about eating broccoli, the high soluble fiber group was eating literally 500 grams of broccoli a day.
Oh,
So
that's nuts.
in terms of the effect on, so alpha diversity went down. That was probably the only thing that was majorly impacted. Lower alpha diversity, but beta diversity, we saw changes in Brachyurtis, but in looking deeper at the genus level, there was some nominally significant bacteria between the groups, but after correcting for multiple comparisons, I mean, nothing stayed significant. So it's just we had a 2x higher total and soluble fiber diet that barely impacted the gut microbiome, that barely impacted fecal short chain fatty acids. And the most important, the primary outcome was fecal pH unchanged between the groups, which is crazy, 2x higher fiber and muscle-related measures. So we looked at grip strength, uh, six, uh, uh, 6 minute walk time and chair stands in 30 seconds. Didn't impact we didn't optimize the gut muscle axis just by exercise, just by diet alone. We had people wear step counters. So we adjusted our models for, because as I mentioned, physical activity can impact short chain fatty acid production, but we adjusted our models for, steps and BMI and sex, but it's a small study, 12 weeks, 20 people, 12 weeks. Is that the answer? Is we didn't get the right fibers for the microbiomes? I don't know.
Yeah, that's quite interesting. Yeah, the other confounder that's in there, as you mentioned, is whether it's, women and men, their gut immune systems have inherently different makeups. My lab is studying that right now, and so but that's the types of immune cells in there, and they're modulated by some of the sex hormones, like androgens, for example, can dampen some types of immune cells in there. Whereas estrogens can promote activation of other types of immune cells. And so that's another interesting way to stratify the data to see if there was any sex specific effects in there. I think would be interesting also. But yeah, that's interesting. I mean, I always tell people to eat their broccoli, but I guess that can't be the only thing you do. But as you said, a whole food diet, there was probably more than just broccoli in there. But and you'd also have polyphenols in there, like in a lot of healthy antioxidants in there. And so, yeah, so I mean, for me, then the next step I would do is I would see if there was a signature that predicted which people produce the most types of short chain fatty acids, if there was a microbiome signature in there.
So the challenge there is 20 people total in the study,
Yeah.
like 9 and 11. So to stratify by sex, almost impossible. I mean, we're comparing three
Yeah.
women against five women. The
Yeah.
thing that's interesting, though, is it had to be a weight maintenance study because we were looking specifically for the effect of soluble fiber, a higher soluble fiber diet. There was no change between the groups, protein intake, fat intake. We matched all of that. But where it gets interesting is we fed a similar diet to aged mice. So when you think about, you know better than anyone, standard chow, it's corn, wheat, and oat
Yeah.
based. But that's the only food. There's some fish meal, whatever that means, in there, but it's essentially a wheat-based diet with nothing else, vitamin mineral mix. So we fed them a quote unquote high soluble fiber diet that was enriched in vegetables, some fruit, I mean, blackberries, we had beets, we had broccoli, all freeze dried. And the animals lost a ton of weight. Like
Yeah.
so I wonder if some of the effects of a high fiber diet, people are going to lose weight. We had to increase calorie intake in the intervention group because they were starting to lose weight and but so that may be a big part of the story where eating more fiber leads you to be leaner and then that contributes to the overall positive health effects in the gut. Whereas if you're eating a high fiber diet and it's just staying, body weight stable, you really don't see any changes in the gut.
Oh, that's a good point. Yeah. So I mean, if the people were eating more calories, to try to keep their weight. Yeah, that might could have had an impact on the microbiome as well there too. I mean that maybe could have diminished some of the effects of having having the extra soluble fiber in there. Um, yeah, that's interesting actually. Um, well, I mean, a lot of so I mean, weight loss, I mean, contributes to, as I mentioned, it contributes to also, it puts like set points in there, but it's hard to maintain some of them. And there's been a lot of studies on showing how the immune system basically, during weight loss will change its migration patterns, and then basically with the weight gain. If there's epigenetic changes that remember that when if you get like obese and insulin resistance, there's like epigenetic patterns in immune cells that remember that. So I mean, I feel like I've seen studies where you're more likely to put the weight back on and become inflamed again based on epigenetic changes in the immune cells. And I think there's been a couple of studies on that. Um, but yeah, no, it's certainly that's possible. But I mean, if you're having more calories and it's healthy calories, that's also, I mean, that's I'm not sure like that. I mean, what would that do to the microbiome, right? I mean, is if you're eating like having too much, broccoli. I mean, there's yeah, what would that do? I mean, you're flooding. I mean, you're getting some of the some of the more like some of the healthy polyphenols and stuff too. So yeah,
So calorie intake
like
between
especially like sulforaphane, right? So if you think of like sulforaphane, right, that's supposed to be like a master. It could be also, if you're eating too many antioxidants. So like sulforaphane is probably one of the famous, antioxidants in broccoli. It's supposed to be an important inducer of like NRF2 and master the antioxidant pathways. But I wonder if this is also an effect. I mean, if you just protect your body too much and you have tons of antioxidants all the time, then it could probably shut down some of your own endogenous antioxidant pathways. So, occasionally I'll take like N-acetylcysteine, right, which is in the glutathione pathway to keep your glutathione levels in check. But if you're basically, filling yourself with antioxidants, it's possible that your own endogenous systems will get lazy when you stop, right? And so, I mean, yeah, maybe it could be something like that. Maybe if you flood too much precursors for too long on your microbiome, maybe over time they stop. But I mean, usually though, in mice, for example, if we give them, a prebiotic or a precursor, basically those bacteria will expand actually. Like the bacteria should be expanding, but I don't know for how long that lasts. And does this expansion become like an exhaustion, like what happens with, immune stimulants? Like if you stimulate the immune system for too long, it'll eventually become exhausted. And I wonder if something like that happens with diet. If you flood, I feel like if you flood your body with too much of one antioxidants, that the endogenous version of that may get tired after a while, or may get lazy, not tired, and then not know how to respond until it gets reactivated again.
Could be. All right. So I've got some mechanistic questions. So tryptophan should be completely absorbed in the small intestine into the blood. So would the indole to tryptophan ratio be a measure of an inefficient tryptophan absorption, as amino acids, their absorption in part decreases during age, or amino acid absorption in part decreases during aging, leading to more of it reaching the large intestine where it can be fermented. So is the indole to tryptophan ratio, could that be used as an index for a somewhat ineffective absorption of tryptophan into the blood?
Yeah, I mean, it's not only the absorption of tryptophan, but it's also I mean, it's possible, as you mentioned, but it's also the metabolism of tryptophan, I think. Um, so tryptophan is metabolized as it goes down the GI tract, but it can also be oxidized, if I'm not mistaken. But it's metabolized by some of those enzymes like IDO and TDO. And usually like the ratio of like indoles to to tryptophan is often, a marker of the microbiome. But as you said, like, a lot of the tryptophan is absorbed or metabolized on the way down, and only a very small amount of it reaches the microbiome. So I think it's about 5% or less of the tryptophan that you eat will eventually make its way to the microbiome. And for me, like an indole versus over tryptophan ratio could be, a measure of tryptophan absorption or metabolism by some of those enzymes along the way, like IDO and TDO, or it could also potentially be like whether you've got the right microbiome in there to produce the indoles. Like if you've got a lot of, like like the right types of lactobacilli or the Clostridium in there, you're going to produce more indoles or beneficial indoles for whatever small amount of tryptophan is left over. So I think it's a combination of two, like how it's absorbed, how it's metabolized, and also the types of bacteria that are in there.
Wouldn't tryptophan degrading bacteria, wouldn't they be more in the proximal colon and less in the small intestine?
The tryptophan degrading bacteria, yeah, so more of those, well, no, the IDO and the TDO, the metabolizing enzymes, are not on the bacteria, they're on the host cells.
But
But
that's not producing indoles. IDO and TDO are producing kynurenine and downstream
that's
metabolites.
right.
Yeah.
Yeah. So, but they're getting metabolized by the host, and those are present along the GI tract. Whereas the indoles are made by the microbiome, the microbiome that produces it, like some of the Clostridium species, and they would be farther downwards, like in the ileum or the colon, a lot of the Clostridium species.
Yeah. So if indole to tryptophan ratio is relatively high in blood, that would suggest that you've got less, no, sorry, wait, indole to tryptophan ratio is high in blood, then you've got more gut bacterial degradation and less tryptophan absorption, right?
Um, well, yeah. Well, you would have more, gut bacterial. Yeah, that's right. Gut
Yeah.
gut bacterial, metabolism of these compounds to the indoles. You'd have more of those.
All right,
Yeah.
so another ratio that I've got in there is the indole to indoxal ratio as a measure of fecal luminal short chain fatty acid levels. So I'm sure you know that the conversion of indole to indoxal is an oxygen dependent process. And so when intestinal epithelial cells, so that's the first step in making indoxal is indole to indoxal is oxidized. So intestinal epithelial cells will uptake short chain fatty acids that'll be used by mitochondria, but consuming oxygen as a part of that process to make ATP in the intestinal epithelial cells. So when the intestinal epithelial cells use oxygen to make energy, to keep that process going, they have to pull oxygen from the intestinal lumen, thereby decreasing the oxygen tension in the intestinal lumen and limiting the conversion of indole to indoxal. So what's core to that process, though, is the short chain fatty acids, because that's what's going to lead to the pulling of oxygen from the intestinal lumen. So what do you think about looking at the indole to indoxal ratio or even indole to indoxal sulfate ratio after adjusting for creatinine levels as a metric of potential metric of fecal short chain fatty acids?
Yeah, so the indoxal levels for these, yeah, don't forget, I mean, well, actually, the sulfonation occurs in the liver, right? So, um, but certainly, I mean, one of the interesting things about short chain fatty acids, and some of the indole compounds, is that there is often some overlap in there. So a lot of the types of bacteria that can produce short chain fatty acids, some of these also can produce some indole metabolites, though to a lesser extent. They're not, like the big ones like the Clostridium sporanges, but some of the lactobacilli that can produce that can produce like indole 3 aldehydes. Some lactobacilli can also produce short chain fatty acids. So there's like, and that's another thing we notice is generally that when you have dietary fiber, um, the dietary fiber. I mean, one thing at least we've noticed in mice, though, when you feed mice with, dietary fiber, not only can, I mean, at least you reduce some of the short chain fatty acids, but you also raise levels of some of the indole molecules. And if you look at, I think there's been some nutrition studies on this where they feed fiber to people. I can't remember the fiber type, but they were able to elicit increases in indole metabolites by giving fiber actually. And so it's possible that relationship is important. I can't comment on the oxygen tension. I have to look more into that, but certainly certainly I think it's possible that short chain fatty acids could correlate with some of the indole productions because of the types of bacteria overlap or work hand in hand while they're being produced.
I could send you the paper that shows, the oxygen tension. I can send you that data. But I'm just interested in, so biomarkers is a big part of what I do, and you can measure some of these indoles with commercially available kits. So there's a metabolomic kit that Michael Snyder at Stanford, Iolo's kit, and they've got plasma levels of indoles, indoxyl sulfate, pericresol sulfate, TMAO. So in theory, you could look at ratios as a metric of, in part, gut health, whether it's short chain fatty acids, anyway. All right, so TMAO, one thing I wanted to expand upon that is I get that red meat has precursors to produce TMA, but what's often I think overlooked as a part of that story is betaine is rich in beets. And ergothioneine, which is found abundantly in mushrooms, can also be converted into TMA. So there's more to the story. I think there's more to the story than it's just, cut down on meat intake. And as a big part of that story is that some of the bacterial enzymes, for example, ergothionase, which converts ergothioneine into TMA, is a pH-dependent enzyme, meaning when it's more alkaline, it's more active. And we're considering that gut alkalinization increases during aging, or on a Western diet, it's going to be more alkaline relative to acidic. So you would have more of this conversion of ergothioneine, and even if it's meats that contain carnitine or choline, into TMA. So what are your thoughts on which goes back to the fecal acidification and plant-based sources of TMA precursors?
Yeah, so I mean, in general, gut acidification, would facilitate, formation of, I mean, at least, I mean, if you have foods that have like, TMAO in there already, gut acidification would definitely facilitate, formation of some of those, more dangerous, N-nitroso compounds. Um, you mentioned, mushrooms. I mean, mushrooms are also, I mean, one of the reasons they're healthy, as I mentioned. I didn't mention this in the talk, but they're also a source of, I think they're one of the richest sources of polyamines, if I remember. Like, and so mushrooms would have, important effects on the gut immune system. And another thing that acidity might do, as you mentioned, like it lowers the growth of some potentially like more pathogenic species in there. But one of the things I didn't get into that much is bile acids as well. Um, and yeah, I'd have to if I remember, I think I saw a study where bile acids are highly they're influenced by the pH of the intestine, and then bile acids, of course, are like major signaling nodes. So yeah, I have to do more reading about how alkalinization and also acidification inside the intestine, contributes to some of those factors, beyond slowing the growth of, potentially pathogenic organisms like salmonella or basically like how they, how it metabolizes, for example. So some of the factors that you mentioned.
Yeah, so ergothioneine, That's another one that's rich in mushrooms. Most people are not eating mushrooms. And plasma levels of ergothioneine decline during aging, whereas TMAO increases during aging. So that could be, where I get that everybody's looking under the meat rock, and I'm not pro-meat or anti-meat. Everybody's looking under the meat rock for TMAO production by the gut microbiome or TMA production. But when considering that ergothioneine declines during aging and TMAO increases during aging, I mean, it could just be a story so I could send you that data too on ergothionease being its activity is maximal pH of 8. And if you look at centenarians, their fecal pH is highest amongst all groups, young, middle-aged, and older. So I
Yeah.
think that's a big part of the story is, and especially for the proteobacteria in the distal colon that are producing things like indoxal or paracresol. So if the amino acids, as you know, if they reach the distal colon, they're going to be fermented by those gut bacteria. And if that region isn't acidified by short chain fatty acids, you're going to get an increased growth of the proteobacteria to make more of these products to put increased, stress on the kidney and all the other things that the uremic metabolites do.
Yeah, I mean, so I know ergothioneine. I think that's sold also as an antioxidant, right? I mean, um, I feel like that's not one of the supplements I've ever tried or taken. But I mean, with I know as you mentioned, it's high in mushrooms. It has antioxidant effects as well. Um, but I mean, for me, when I think mushrooms, I usually think polyamines as well. I mean, that's one of the things I've always thought with mushrooms is also a highly rich source of. I think it's one of the highest sources of some of the polyamines in there.
You've also got fiber, you've got niacin that can be converted potentially into NAD and all of that, good stuff too. So, all right, I think I could keep you on here forever, Dan, because I have a ton more questions. But that was great, and I'm looking forward to hosting you again at some point in the future on the channel.
Oh yeah, thanks so much. Yeah, thanks for having me. Yeah, I mean, yeah, had a good time. Yeah, send me some of those papers that you talked about. I haven't looked too much at, colon acidification, um, in my research, but I mean, people, especially on immune cells, are looking at, how acidification, at least in terms of metabolic byproducts, impacts, immune cell function. And so like I'd be interested in that from that regard as well. Um, but yeah, no, thanks. I learned a lot from you too, and yeah, it was a nice conversation, and yeah, I look forward to speaking again on something else maybe.
The B cell T cell story is a huge story that,
Oh
so
yeah, yeah, I can present that. I'm presenting that at the in June at the what's it called? The Gordon Conference. I'm doing that one in 2 weeks, so I'll present that. But happy to present something for you guys here too if you want. I mean, that
yeah.
one has been mostly in mice though. But I mean the data was profound. I mean, we had one of the longest lived genetic knockdown mice ever described in mice lacking B cells, and it was dramatic and highly reproducible. And so, yeah, I mean, I'm happy to do a short one on that if you want. I can do
Yeah,
that.
yeah, definitely. Yeah, just as a quick question there. So, when considering that lower B cells may optimize, I think it was CD4 cells in your study.
Yeah,
Yeah, so that calorie restriction reduces B cell levels, which suggests that, maybe that's
right.
one of the reasons why calorie restriction leads to dramatically extended longevity is by reducing B cells, takes off the suppressive effects on CD4 T cells. And just to add this to a little bit too, I don't know if you saw a lesson.
Before you say, I put that in my grant rebuttal because people were asking me about that, and I found that paper and with the calorie restriction. So that paper I know, and that one, I mean, they gave two levels of calorie restriction. They gave a mild restriction and a more significant. And when you give the more significant levels of calorie restriction, the B cell levels just kind of, fell off a cliff and started going down pretty fast. And that's what I thought. I put that in the rebuttal of my grant. I'm like, yeah, that's probably one of the reasons, why calorie restriction is, is improving, the T cell repertoire. It's possibly acting through B cells. So yeah, 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 that study, I know that one.
Hopefully the grant reviewers like that because sometimes grant reviewers, you know how it goes with grant reviewers. So.
I know, but yeah, I mean, hopefully, but yeah, that one, yeah. Sorry, I didn't want to cut you off. You were going, yeah.
No, so Alessio Lana's group, it's a preprint, but they've been studying CD4 T cells in antigen presenting cells, and basically, um, I don't know. Have you seen that work? The paper is called. It's basically built around telomere rivers, where if you the
Uh,
antigen, have you seen that? Yeah. So that
yeah.
not a fan of that
No, no, no. Is that paper out yet? I think I saw a preprint or something of
preprint.
that.
Preprint. Yeah, his preprints. He said it takes like 4 years for the reviewers to finally approve it, which is crazy.
Yeah, I want to know if that paper ended up getting accepted because they somebody at I don't know if I should say this. I was asked to review it, but I was too busy at the time, and so I did not review the that paper because I was way too busy. I was like I couldn't remember what I was doing, but I saw and then I looked at the preprint and I saw it and I that was they had like this remarkable massive improvement in longevity way beyond anything that's ever been reported in mice
Yeah.
and it I mean normally if you have a good effect or which has been consistent with the literature with a drug or something is maybe like a 10% increase in lifespan. Genetic models you can go all the way up to like 30-35% if something is dramatic, right? I mean our B cell deficient mice had like a 35% increased lifespan in females and that was one of the largest I've ever seen. And then this telomere river study had a it was like the mice were living 5 years or something crazy
Yeah.
like that, weren't they? It was insane. I mean, black six mice living 5 years. I didn't even know if that was possible, but I guess it is if they've reported it.
Yeah. All right. So that's just a small snapshot. Hopefully we dive all into
Yeah,
that, for the next one. So, all right. Thanks, Dan. I appreciate you and
yeah,
have a great day.
yeah. I just want to check one fast thing. Because I just want to see there is that lactate. I just want to see. I'm just going to Google this now because I know the author who did the lactate study. Um, yeah, so that's right. So basically, there is a lactate study here, and there's two types of lactate. There's a D-lactate and an L-lactate. So and I'm just looking at this on Google right now. So the D-lactate is produced by the gut microbiome, and L-lactate is produced by exercising muscle, and it's basically the D-lactate that's produced by the gut microbiome. Which can actually act on the liver to cause it to raise inflammation and raise glucose. So there's two forms of lactate. It's D and L lactate. And so it's the lactate specifically made by the microbiome, which is the one that's inflammatory. Yeah, so
What's
I knew there was a study, I just couldn't remember. And obviously this also contributes to acidification of the gut. I mean, when you have more, lactic acid in there.
what
But yeah, go ahead.
what's the prebiotic for those bacteria to make lactate? Would it
Yeah, I have to
be
look at the paper. Let me
yeah,
check. I just Googled it just now.
if pyruvate, that would suggest that anything that produces pyruvate, like exercise training, would be bad for the gut, which would be the opposite of what I would expect,
But
though.
but like the muscles like from exercise training make L-lactate, which is a different isomer of lactate, and that does not seem to aggravate inflammation. It's the D-lactate produced by the gut microbiome, and I have to look at I don't I'd have to look at the paper to see how it's actually producing.
Yeah.
I mean, I can put the paper over here if you're
Yeah,
interested.
yeah, yeah. I'm
Uh,
going to do a deep dive and look into D-lactate production by the gut microbiome. This is great stuff.
yeah. Let me share this paper right here. Yeah, sorry. Yeah, it's yeah. As I was saying, I had a little bit of like brain fog 'cause I'm like, my nose is running still a bit from the virus I had last
Ah,
week,
cool.
but yeah, so but I remember I specifically saw a paper like this, right? So there it is. Yeah, then there's two forms of lactate, which can and it's the D-lactate, which can, acidify. But as you said, acidification is usually not so bad in the colon because it reduces, some of the dangerous Clostridium or Salmonella or whatnot. Um, but but at the same time, if it's, D-lactate if this is lactic acid if this is forming lactic acid, it's going to acidify the colon, and probably this gets into circulation too and causes some inflammation, it looks like.
Yeah, good
Yeah,
stuff.
so
All
those
right.
are the
Thanks,
pictures.
Dan. Talk to
Yeah.
you soon.
Okay.
All
Yeah. All right.
right.
Take care. Bye.
Bye.
Bye.